Okay, let's dive right into this explainer. We're looking at a real-time biological thriller playing out in the poultry farms of Egypt. Basically, a rapidly mutating virus is threatening the very foundation of the country's agricultural food security. And honestly, while this is unfolding in the Nile Delta, the lessons we're seeing here about viral evolution and vaccine pressure, they are absolutely crucial for global health everywhere. It's a fascinating, if a bit terrifying dynamic.
So, here's our roadmap for today. First, we'll look at the Egyptian poultry crisis itself, then examine this shape-shifting viral threat. After that, we'll see how it goes way beyond just respiratory disease, uncover a deadly alliance with H9N2, piece together the diagnostic puzzle, and finally look at strategies for flock survival.
All right, let's get into it. Section 1. The Egyptian Poultry Crisis.
Now, when a highly contagious pathogen infiltrates the densely packed poultry farms of the Nile Delta, the stakes are literally staggering. You see, Egypt's massive poultry sector mixes these huge commercial operations right alongside widespread backyard farming, which creates just the perfect storm for transmission. The crucial point here is the mortality rate. We're talking about a terrifying leap past 50%. In an isolated, stress-free lab environment, this virus might just cause a mild cough. But out there in the real world, throw in dense flocks, poor ventilation, and co-infections, and it becomes an absolute catastrophic killer.
To really understand the villain of our story, we need to define IBV, or infectious bronchitis virus. This is an incredibly agile RNA coronavirus that uses a heavily glycosated spike protein to attach to and invade host cells. Just like the coronaviruses we're so familiar with in human medicine, IBV relies on that exact spike protein to breach the host's defenses. But remember, IBV has had decades to perfect its infiltration techniques specifically in avian populations.
Moving on to Section 2. A shape-shifting viral threat.
You might be wondering, why is this virus such a nightmare to control? Well, it's the ultimate biological shapeshifter. It's constantly rewriting its own genetic code to stay one step ahead. It essentially evades immunity through a three-step survival strategy. Step 1. Sloppy proofreading. Its genetic polymerase makes constant point mutations in the S1 gene. Step 2. Glycan shielding. It literally attaches molecular sugars to itself to physically hide from the bird's antibodies. And step 3, genetic recombination. If two different strains infect the exact same cell, they can actually switch tracks and swap parts, creating a completely new Frankenstein-style chimeric variant.
When we look at the circulating genotypes here, you can see the classical strains GI1 have been pretty much completely displaced by the aggressive GI23 lineage and a newly emerging GI19 threat in Upper Egypt. GI23, also known as variant 2, is hyperendemic. It is the undisputed heavyweight champion right now, causing huge economic losses. But the emergence of GI19, or the QX-like strain, is really setting off alarm bells for researchers, because it brings a whole new set of devastating clinical impacts.
Which brings us to Section 3, Beyond Respiratory Disease.
The name infectious bronchitis is actually kind of deceptive, because this virus goes way, way beyond the lungs. This broadened tissue tropism means it doesn't just cause gasping or severe ciliary destruction, it literally liquefies kidneys, causing fatal uremia, a condition known as spotted kidneys. It permanently destroys reproductive tracts, creating what's called false layer syndrome, where you see a massive 50% egg drop. It hits the digestive system with proventriculitis, and it severely suppresses the immune system through lymphoid depletion, practically rolling out the red carpet for secondary bacteria.
And that leads directly into Section 4, the deadly H9N2 alliance.
This right here is the climax of our story. We're looking at a horrifying biological synergy where our shape-shifting villain meets its deadly accomplice, the low pathogenic avian influenza subtype H9N2. Now look at the difference. While either IBV or H9N2 alone is a minor nuisance with near 0% mortality in stress-free settings, together, they trigger a massive, dysregulated hyperinflammation. It's a tragic math equation where 1 + 1 = 1,000. The host's immune system just panics, overreacts, and essentially destroys its own tissues, sending mortality skyrocketing past that 50% mark.
Okay, let's dive into the molecular mechanics of how this actually works. The IBV infection fundamentally alters the bird's biochemistry. It physically unlocks H9N2's infectivity and extends its viral shedding for days. You see, the influenza virus needs host enzymes to cut or cleave its surface proteins, the hemagglutinin, to enter cells. Usually, it's restricted to areas where those enzymes naturally exist.
But IBV acts like a master skeleton key. It provides the exact protease enzymes that H9N2 needs to spread unchecked throughout the entire body.
This synergistic alliance just strips away all physical and immunological defenses. It starts with the destruction of the mucociliary escalator in the throat, followed by that massive cytokine storm we mentioned. Airway occlusion happens as dense bronchial casts form. This creates an undefended superhighway for environmental bacteria, specifically avian pathogenic E. coli, to sweep in unhindered. And it's actually the secondary bacterial superinfection that pushes the host to ultimate failure via fibrinous polycerasitis, delivering the final fatal blow.
Section 5. Solving the diagnostic puzzle.
Because symptoms look identical to highly pathogenic bird flu or Newcastle disease, virologists really have to play detective to crack this complex case. Out in the field, you simply cannot tell what's killing the flock just by looking at them. Traditional serology, like ELISA or HI blood tests, they're effectively useless here. They get completely obscured by high vaccine background titers and are prone to false negatives due to viral drift. That makes the molecular gold standard, real-time PCR detecting the conserved N gene and full S1 sequencing to identify the exact variant, literally the only ways to definitively unmask the virus.
But even the gold standard has its pitfalls. Our lab detectives have to navigate some serious hurdles. First, vaccine interference. Live emergency vaccines cause massive false positives in the lab results. Then there's multiplex competition, where the fast-replicating IBV can actually hide the slower H9N2 in the exact same test tube. And finally, sample degradation. Imagine trying to sequence a delicate viral genome on an FTA transport card that's been sitting in a truck in 40 degrees Celsius Egyptian summer heat for 2 weeks. The data literally melts away before it ever reaches the lab.
All right, let's look at the counterattack. Section 6. Strategies for flock survival.
To break this cycle of continuous viral evolution, the industry requires some highly strategic interventions. To prevent catastrophic vaccine failures, they rely on two main approaches. First, homologous vaccines. These are custom matched to the circulating strain, like GI23, kind of like a targeted sniper rifle. They provide 80 to 100% clinical protection and stop viral shedding. The second approach is using heterologous protectotypes. This is more of a shotgun approach, combining distinct classic and variant live vaccines to elicit a broad, cross-protective mucosal immunity.
And this is absolutely fascinating, we face a massive paradox with these interventions. Flooding the environment with diverse live vaccines inadvertently provides the exact biological substrate required for genetic recombination. Think about it. When a bird is simultaneously replicating highly divergent vaccine strains, it becomes a biological mixing bowl. The virus can literally swap genes with the very vaccines designed to stop it, birthing a brand new elusive field variant. So it leaves us with this chilling realization to ponder. As our agricultural vaccines push viruses to evolve faster and become more elusive, who is really winning the biological arms race? Are our most advanced tools for survival actually training these pathogens to become the perfect biological villains? Understanding this delicate balance is absolutely crucial, not just for Egypt's agricultural economy, but as a profound lesson in how we manage viral ecosystems globally.